Good morning, everybody. Do myself comfy. Whew. All right. Sorry, bit of a rush. I was working on my wife's Christmas present for class. It's going to be a giant metal chicken. All right. Bring up the chat. All right. So we finished up Drip Operon on Friday. Now, I know it's Friday and today's Monday, which isn't a great combination. But does anyone have... let me just get back to where I was at. There we go. I can see the question. No, you've got it completely right. So that's the first, I don't know, concept, I guess, right? So and don't forget it's the leader region as well. Let's look at everything. Yeah. So that's the leader region that you're talking about, which is, crabs, where is that? Nope. Nope. Badgers. There we go. I knew it was in there somewhere. So that leader region is a short bit of mRNA that's part of the polycistronic mRNA, right? So it's part of the large piece of mRNA that encodes everything for the trip operon. And that gets transcribed first, right? So the fact that it can form two different conformations is, there we go. Really important to how this works. Right, and this isn't, this isn't entirely correct because this 1-2 loop doesn't actually form. It's either the 3-4 or the 2-3. So those are the two possible conformations, right, of that leader mRNA. Now, question to you. What dictates which one of these conformations form? How is the level of tryptophan actually sensed by this system? Trip codons, how? That's correct. What's the next part of the picture? So we have trip codons in region 1, right? Those are kind of, in a sense, part of the detector. So how do these play a role in these two different conformations? Why are they important? How do they do it? Correct, right? So, okay, so let's kind of go back a little bit, right? So what is one of the key characteristics of gene expression in prokaryotes? Very different from eukaryotes. What's the characteristic that allows all this to work? Right, awesome, Isaiah. Transcription and translation occurs at the same time, right? They're coupled. Obviously, transcription is a little bit ahead of translation because you need mRNA to translate something, but they occur together. Okay, so two different conformations, 3-4. This one. Go back. Sorry, picked up the wrong thing. Or this one. Right? We have, get ahead of it. Sorry. That'll be fine. Two different conformations. We've got trip codons which detect the level of tryptophan. How is that level of tryptophan detected? Right, so if you have tryptophan present, what gets incorporated? If you don't have tryptophan present, what does not get incorporated into what? Right, what's being made as this leader sequence is being transcribed? What's being made? Again, going back to what Isaiah wrote up in the chat. We're making mRNA, obviously, right? RNA pol is kind of somewhat ahead. What's being made at the same time as the mRNA? As soon as enough of it appears for this to start. It uses tryptophan, right? So what is made up of amino acids, which tryptophan is one of? Proteins, right? So this, yeah, right? So this is where that comes important, right? This is where that simultaneous or coupling of transcription and translation is so important in this particular system, right? It's not really so much in, oh, I mean, don't worry about it, Melody, just answer out. If you don't get it right, it doesn't matter. It means you learn something by getting it wrong. If you do get it right, then you go, well done, and you feel good about yourself. I mean, you feel good anyway, right? Oof, tying myself in knots. I would never make a good psychologist or psychoanalyst, to be honest. I'd always be putting my foot in my mouth. Anyway, so we're making protein. Protein needs tryptophan, right? Why does it need tryptophan? Because the trip codons are here. Okay, so now we've got to hold a lot of things in our mind at the same time, right? Two different conformations. One of which will occur if tryptophan is present, one of which will not. Okay, so which one will occur if tryptophan is not present? So think about what the ribosome is doing and where it's at. Right, and why, Stephanie? Why will the 2,3 conformation form, or the option form, if tryptophan is absent? You're 100% correct. All right, the ribosome gets stuck where? Exactly. Good, awesome. You're getting it. That's really cool. So if the ribosome pauses on region 1, region 2 is going to get transcribed, and then region 3 is going to get transcribed. And so if you have region 2 and 3, what are you going to get a loop formed between? 2 and 3, right? Because 4 hasn't been produced yet, right? This is why it's a linear process, right? So we have two conformations, possibilities, I guess one could call it. We've got trip codons in region 1, and translation is occurring at the same time. So let's go down to here rather than me waving my fingers around. If the ribosome stalls, because there's no tryptophan, right? No tryptophan equals nothing to go into those trip codons, right? Then region 2 and 3 are free to form this hairpin loop. And that hairpin loop has no effect on transcription, because RNA polymerase is already kind of way ahead of the game before this forms. It's already transcribing region 4. Okay, how's that go? Does that make sense? You got to keep a lot of things kind of up in the air in your mind at once. Any questions? The ribosome, we don't care about the ribosome, actually, is the answer to that. The RNA polymerase, I don't know if you meant RNA polymerase instead of ribosome. The RNA polymerase is too far away. So remember, we need, where do we have a good picture of that? Back up here, I guess. Right, we need these poly U's, that's right, because this protein that is being made has no purpose. We don't care about it. Right, it is simply there to allow the level of tryptophan to be detected. So this whole leader region is really I mean, it's essentially part of the regulatory region, right? Oh, that's okay. Good that you asked them. We don't care about the ribosome. We don't care about that protein. It's merely being made as a means to detect tryptophan. And, you know, what better way of detecting amino acid levels than to try and make a protein using that amino acid, right? I mean, it's pretty elegant if you think about it. So let's go back. Now we have this poly U, right? And so which hairpin loop forms, whether it's 2, 3, or 3, 4, depends on where the ribosome is, right? Because the ribosome essentially dictates which ones can form. If the ribosome is on region 1, region 2 and 3 will form, right? And it'll only be there if there's not enough tRNA to synthesize the protein, right? Now, what will happen if the ribosome moves through and it moves into region 2? Can region 2 and 3 form a hairpin loop if the ribosome is on region 2? That's right. Region 2 and 3 can't form. So 3 basically is unpaired, right? This is kind of the key, because remember, this is a linear process, right? Each region kind of gets made in turn. And so as soon as they appear, if there's one that they can pair with, they'll pair with it. And so if region 2 is covered up, when region 3 is being made, region 3 can't pair with it. It's just kind of just hanging around, right? And then region 4 is made and the RNA pol pauses on these U's. And then we get this terminator loop form. And you can see how close it is to the U's, right? Versus region 2 and 3, right? Region 2 and 3 is like a fair distance away. Region 3 and 4, when they form, are right next to those U's. So this is occurring inside the RNA polymerase. Right, and so you see that here, and because that forms inside the RNA polymerase, it kicks the RNA polymerase off. So essentially, even though the RNA pol was able to start transcription, right, back over on the left, because there's no TRIP-R bound to the operator, right? So there's not like crazy high levels of tryptophan, which would just shut everything down completely. Right, there's still like low enough levels that transcription starts, but there's sufficient tryptophan for transcription to stop. And it's stopped before it gets into, there's that picture that I'm after. Yeah, it's stopped before it gets into the structural genes. Right, which starts kind of over here, essentially. How about now? Cool. I'm glad you brought up about the ribosome. I should have mentioned that originally. It's just sometimes it falls through the cracks. Cool. Does anybody else have any questions? So when you encounter, questions about this, which, obviously you will do in the exam, work through what is going on, right? Again, it's like the turtle versus the hare kind of deal, tortoise maybe. I'm sure which one it is. If you work through it, kind of logically, you'll get to the right answer. Cool. All right, well, if no one has any questions, we can move on. Let's see what is next. Ooh-hoo-hoo. Okay, so in many ways, this one's the big one. All right, so there's a lot going on here, right? And so in eukaryotes, let me kind of type this down. So let's state what we already know, right? Prokaryotes coordinate gene expression by grouping structural genes into operons. Right? And polycistronic mRNAs as well, by extension, essentially. Gene expression is on by default in prokaryotes. Right, so it has to be turned off, right? And it could be off kind of automatically, right? Which is what the lac operon is, right? And it's off because of the presence of the LAC-I repressor, or it could be repressible. Actually, let's just type this out rather than telling you like this, right? Can be off due to the presence of a repressor, which must be induced to leave, inducible, e.g. the lac operon. And this is all in prokaryotes, okay? Can be on and repressed by an apo, sorry, gotta get my grammar right, and co-repressor. And that is repressible, e.g. the trip operon. Oops. So now you can see why we use those two as examples, right? So they're just really good examples of both. And also, as you can see, most, maybe not quite all, I couldn't tell you for certainty, gene expression in procs is, and that's short for prokaryotes because I'm lazy, is controlled at the level of transcription. Okay, so that's prokaryotes, right? Eukaryotes are much more complex, massively so, in fact. And in many ways, that complexity in the control of gene expression kind of is reflected by the complexity in organismal shape and form, essentially, right? I mean, Yeah, I guess even protists, and yeast and things like that. If you look at the life cycle of malaria, right, for example, it's astonishingly complex, right? Even though malaria is a single cell organism, a parasite, it goes through like 4 or 5 different forms. They've all got like funky names like schizonts, stuff like that. I'm not much for a malaria parasitologist. Anyway, even single cell level, there is an astonishing complexity of form, right? And that is the result of many different levels of control of gene expression, right? So in many ways, and this is where a lot of the complexity comes in, the big deal about eukaryote gene or control, sorry, of gene expression are the varying scales. Or in other words, all the different levels at which the final outcome, I guess, which is by and large functional protein, not always, obviously, functional mRNA or mRNA, functional RNA, is another functional outcome, essentially. And so there are a lot of different ways, right? And so another way of looking at it is that gene expression in Ukes is coordinated, right? Obviously, if you're often use developmental biology examples here because it's kind of easier to grasp. If you're making an arm, which I have 2, as do I think all of you, you need to express all of the genes, most likely in a particular order, that are required for making an arm, right? And so arms start off as, they're called limb buds, super cute. Makes like a little, what are those kind of like sea-born dinosaur things, like the big flaps, paddle things. Anyway, it's kind of like that. So they start off as a limb bud, right? And that extends out, and as it extends out, you lay down or differentiate the different parts of your arm, right? So you get whatever that bone is called. Yeah, I can't remember. And then you get an elbow joint, then you get these 2 bones, and then finally you get, your hand. And so each of those steps requires different proteins to be expressed, or different genes to be expressed, different things to be made, right? So gene expression in eukaryotes is coordinated. I'm going to put emphasis on is. But in many different ways, right? And in some ways, this is both the topic I like in talking about the most, right? Because really, this is where we get to put together all of the cool stuff that we've learned, right? And this is really, in many ways, it's kind of why we are the way we are, right? As an organism, but even at a cellular level, it's why our cells are what they are, right? It's also one of the hardest topics to teach because it is so astonishingly complex, right? So I've tried to simplify it as best I can, but try and keep in mind like the big picture, right? Complexity. Okay, and so, have I shown you the alligator embryo picture yet? Ah, oh, you can also look at my cat and dog pic folder. Anyway, let's get it out of the way. Boop-doo. That's work. You get like a nice little, you can look at my kitty cats, some foster pups, it's a friend cat wearing a sombrero, all kinds of fun stuff. Here we go. Got there eventually. I love this picture. It is just fabulous. So this is a developing alligator embryo. Right. I don't know what stage it's at. I'm not much of an anatomy and physiologist, to be honest. Right. So the actual kind of like what things do and stuff, not so good at. But this is awesome. This is so I'm a kind of microscopist, I guess, as well as a geneticist. So I love microscopes, taking pictures of small things, seeing things you can't normally see. So this is a fabulous image. Actually, do I have it in the PowerPoint somewhere? Let me just double check. I want to see if I've no, I didn't. That's okay. Anyway, so this is what's called a confocal laser scanning microscope image. And so essentially this is taken by scanning a laser, a very fixed depth across the sample. So this picture probably would have taken, all told, probably a couple of days to take, I would guess, because this is a fairly big thing, you know, in microscope terms, at least. You know, I'd imagine it would probably be, you know, at least kind of that sort of size, right? Which is bloody huge when it comes to microscopes. And that laser is at a very specific frequency. So you remember me talking about fluorophores, right, in lab when we talk about Sanger sequencing, like the different colors that are used to label the different A's, C's, T's, and G's. All right, it's the same deal here. It's a very specific wavelength, which will excite one of these colors. And then it does it all over again for a different wavelength, which will bring out a different color, right? Which will excite a different fluorophore. And it does that at layers through the sample, right? Because this is a relatively thick sample too, right? Probably, I'd guess, where's the camera? About that thick, give or take, right? Which again is huge, microscopy wise. I have issues taking pictures of worms, which are about 50 microns thick. So a micron, 50,000th of a 50 millionth of a meter, whatever that is. Small, right? This is much bigger than that. Anyway, so those are all just kind of smooshed together in a program, and then the different colors are overlaid, which will give us this fabulous picture. And so essentially, you look like real close, you'll see this is the developing brain of so all the green stuff are nerves, neurons. I should actually put this in the PowerPoint. That would be a better idea, wouldn't it? Right, so you can see all along here, this is essentially going to become the spinal cord, this line along the back here, right? And these are probably different areas of the brain. Even though this looks like the head, it's not actually complete yet. And so these are called the branchial arches, which will come together and actually form the jaw. So this is the same in humans as well, right? So your jaw has actually got a joint line, essentially. This is where hair lips come from, right? Cleft palates, right? And so kind of about up to here, like eyes and above is part of what you see here, this bobbly bit sticking out. Kind of cheekbones and below is formed by these brachial arches, which come together. It's really kind of alien-like, right? They kind of stick together like that. And that's what makes your lower jaw, your upper and lower jaw, and teeth and things like that. So muscle is going to be in orange by and large, right? So this is the developing heart. These are blocks of tissue. These are called somites, right? And these will differentiate into back muscle and vertebra, right? And so this essentially is the mark of a vertebrate, right? Is this formation of somites, these kind of groups of distinct, different bits of tissue. Now, each of these somites are discrete, right? They are separate from each other and they have a different identity. And from that different identity, you get arms and all kinds of funky stuff. You get liver, you know, where you should have a liver. You get kidneys where you should have kidneys, right? And so all of this comes from the same set of instructions, right? So you can see even just in a developing embryo, right, the astonishing level of difference, right? And yet all of these different tissues, every single one of them, in fact, has the same genetic information, the same instruction set, essentially. And yet, as you can see, even in this unformed state, they have extraordinarily different roles, morphology, location, right? So the heart is only here, right? And even though these are also future muscle and bone tissues, is kind of what we call mesoderm, pardon me, they're in a very different place and they'll become very different things. And so all of that is the result of differences in gene expression. Now, obviously, looking at this is far more complex than a bacterium, right? One of the reasons why I work on eukaryotes, right? I find even though bacteria are super, super cool and they have lots of like really neat ways of doing things, there's just so much more fun to play with, right? More stuff to look at, right, with eukaryotes. And so all of this is, even though it's not yet finished, obviously, because it's not something that will, you know, grab your puppy and drag it to the bottom of the swamp in Florida, right? Even so, right, we have all of this crazy differences in gene expression, right? How cool is that? It's just such a fabulous image. I've got a link to it somewhere. I don't have it in this PowerPoint, but I'll dig it out so you can go read about that. We'll do that later after class. And so really, it's all about the different scales, right? So this is just a cool image. We'll come back to this. This is only one part of the control of gene expression in eukaryotes. This is a control of transcription, which is admittedly a very, very, very big part of gene regulation, but it's only one part, right? And so what I really want to talk about, and this will take, I will probably spend the next, 15 minutes or so until the end of class talking about this, are the different levels at which regulation can occur, right? So essentially, I mean, cells can take instruction from within, right? An intrinsic signal, like change in nutrients level or energy state or, cell health, I guess, however that would be measured. Or they could have a signal from outside, an extrinsic signal, right? And that's in many ways what tells different cells to become different things, right? That's why, your heart cells are different from your heart muscle cells are different from your arm muscle cells, right? Because they've been told to develop differently, even though they're both muscle. So quite often an external signal is involved, right? Anyway, first level of complexity is at the level of chromatin, right? And so, oh wow, where did we talk about that? I'm just trying to think. No, that's not in that one. That was a way back. Hang on. See if I can find it. Oh, I don't know what I just opened. Oh, the signal. The signal could be all kinds of different things, right? So cell signaling is a very complex topic all of its own. I'll find that another day. So it could be a protein, right? Could be like insulin, for example, is an external signal that tells muscle and liver cells to put a glucose transporter into their membranes. Could be something like retinoic acid, right, which is a metabolite. Actually, I don't know if any of you have ever had treatment for acne. I think it's, what's it called? Retinoin? Something like that, right? It's a very, very dangerous chemical because if you have it or you're taking that particular treatment for like really bad acne while you're pregnant, you could very much mess up the development of your unborn child, right? And so there's a whole bunch of stuff kind of going with that. It could be lots of different things, right? So it's essentially some piece of information from outside the cell that the cell needs to kind of hear or see in anthropomorphic terms to make a change in its gene expression, right? So it originates outside the cell and either gets detected by receptors on the surface or actually travels through the cell to the nucleus and affects gene expression there. So that's just one enormous other kind of field of topic. I don't even get to talk about all of the different ways that happens in my Dev Bio course, which is in large part about cell signaling. So the very first level of change is about chromatin, right? So remember that we talked about euchromatin versus heterochromatin. If I could spell that right. Which is essentially open versus closed, or on versus off, right? And so that's carried out by a lot of different means, right? That switch between on versus off DNA structure. And that can affect really large parts of a chromosome at once. And so for a lot of these genes that are involved in these developmental programs, for example, they're actually found kind of grouped together. And so if you don't want to grow any more arms, which, you know, I don't, I've got plenty enough for what I need to do, then you can shut down that whole program together by changing its chromatin state, right? So that's kind of like a big scale level, right? Many different genes, large parts of the chromosome. And if you think about it, we've got 20,000 genes. I'm not entirely sure how many are being transcribed at once, but I think it's probably in the 2,000 to 3,000 range. So a large majority of the genes that you have in your genome, every single one of your cells, are off at any point in time, right? And one of the ways in which that is done is through the formation of heterochromatin, right? And it's a dynamic deal, right? It can switch back between one and another depending on need. So once you have euchromatin, open, accessible chromatin, then that DNA is now available for transcription if the correct proteins are present, right? So remember, gene expression oops, in Ukes is off by default and needs something, usually a protein, to turn it on. Right, so if those proteins are present, transcription factors is their kind of generic name. Proteins that turn on gene expression or transcription, sorry, got to be careful here, are called transcription factors. Right. So transcription occurs, you have your primary transcript, right? Then that's not, you know, RNA that can be translated yet because it needs to be processed. Again, here's, along with this chromatin formation and dynamic changing between heterochromatin and euchromatin, RNA processing is another difference between prokaryotes and eukaryotes, right? So bacteria don't have chromatin, they don't process their mRNAs because their mRNAs don't have introns. Okay, so those are kind of two pretty fundamental differences. That mRNA is transported to the cytoplasm. I don't think there's any particular level of control there that I'm aware of, but obviously there may be. And then obviously we've got translation, and that also can be controlled, right? How often translation occurs, what frequency, how strong translation is, and also kind of sort of related to that, like a yin yang kind of teeter-totter seesaw thing, is how stable the mRNA is, right? So not only can you control the production of protein or polypeptide, really, because we're not quite there yet, by controlling translation, you can also control it by essentially dictating whether or not there's any mRNA there to translate, right? So these two things, control of translation and control of RNA or mRNA stability, both have the same outcome, which is a control of how much polypeptide is produced, right? And that's controlled as well. It's even the degradation of mRNA, which is like, whew, yeah, I like scratched at the surface of that several times. I actually used this as a technique in my research, but yeah, I tend not to wander into those particular wildernesses too often. So even once we have the polypeptide, okay, we don't have functional protein yet. Right, this is the crazy thing, right? We've made protein, but we don't have functional protein, right? And so, oh, I work on, actually, funny enough, cell signaling. I use C. elegans to look at the effect of overactive growth signaling on a variety of different things. Currently on lipid synthesis, right? So lipid metabolism, also on cell survival, and I've got some other stuff in the wings on innate immunity, right? So kind of worms' ability to fight off bacteria. Oh, and also some developmental biology stuff too. So a bunch of different things, all kind of centered around growth factor signaling, which is kind of why I often use those sorts of examples in talking about genetics, because I'm pretty familiar with them. So anyway, like oh, thank you. I like it. It's fun. Keeps me out of trouble. Where was that? Yeah, so like the general kind of idea of gene expression, you know, sequence hypothesis or central dogma, if you will, right, is DNA, mRNA, protein, right? And so there's the idea that as soon as protein is made, now it does whatever it's supposed to do. And that's not true. It's not like wrong, it's just not the whole picture, right? You take that translation bit and you expand it, translation, processing, export, then at the end you get functional protein, right? And so essentially in many ways that's what we're doing. We're taking this fairly simple idea of gene expression and now we're expanding to look at all of the different bits kind of in there. And so even after translation, we have this polypeptide, right? But then that protein needs to be folded correctly, which is not a trivial deal if you think about how big and complex these structures are. It typically, most certainly will be processed in some way, such as by cleavage, like snipping bits of it off for it to be active, or adding things, to different amino acid residues to get our active protein. And now that, depending on what protein it is, right? If it's just in the cytoplasm and just hangs out there, does stuff. Maybe that's the end of the kind of list of things to talk about. But other proteins are required in different places, different cellular compartments, right? So we've got all kinds of organelles in a eukaryotic cell. Some of these proteins have to go back into the nucleus, right? What about DNA polymerase? It's not translated in the nucleus. The mRNA that encodes the DNA polymerase protein is exported into cytoplasm, translated into DNA polymerase, but then it has to get back into the nucleus to be able to actually synthesize DNA, right? Some proteins end up on the surface of cells, like those receptors that I told you about. If you think about any of the proteins involved in immune function, they're all on the outside of the immune cells, right? Some proteins even just get kind of punted off out into cellular space. That's a bad joke. And wander around doing other stuff, hormones, insulin, right? When I jump out one of my kids and give them a fright, not that I do that because that was obviously a terrible thing to do as a dad. They'll dump a whole load of adrenaline out of their adrenal glands and go, ah, and either like smack me or run away. Maybe. Those are all hypothetical situations, obviously. So those proteins have to travel around the whole body to be able to do their job, right? And so clearly, until they're in position to do that, they're not functional. And also how long those proteins hang around for, much the same as how long mRNA hangs around for, right? Determines whether or not we get translation. How long those proteins hang around for determines how long we get function. Pardon me. And some proteins are turned over pretty quickly. Cell signaling proteins, for example, as part of their function, get destroyed once they've done their job. Others hang around for a long time, but even those will get, beat up and broken and become non-functional, right? So they need to then be degraded and recycled and then used to make more protein, right? So really, this is like one of those complex equations, like, probability of life elsewhere in the solar, like the galaxy kind of thing. Every single step of the way, there is a potential ability to control the final product. So the final product, we might start with chromatin over here, DNA in its more complex form, and we have functional protein at the end. That is the sum of all of those steps. And so now you can see how complex this is. And on that note, I'm going to take dogs for a walk, have a cup of coffee, and make some more of my metal chicken. Cool. Oh, when it's looking more like a chicken, I'll show you what it looks like. And you can either be amazed or entertained, one of the two. Cool. All right, everybody. Thanks very much. Cool questions. If you have any questions about any of this, just bring them to next class and we'll cover them then. Yeah, have a great day too.